All right, so yeah, that was a great introduction by Derek. And so today I will be talking to you about some of the research from my PhD thesis, which was very recently published in December last year. And we were looking at magic beaks, the sixth sense in birds that dates back to the time of the dinosaurs. Sorry, I've got a bit of asthma, so I might just pause every now and then to drink some water, but we should be good. So yeah, this project was done with my supervisors, Anusia Chansami and Susan Cunningham, who, as Derek said, will also be giving talks later on this month, and they're definitely worth attending. They're both wonderful women in science and great bird lovers as well. So firstly, the birds that I'm going to be talking to you about today are part of a group or part of a foraging guild known as probe foragers, and this refers to their ability to catch hidden or buried prey. Anyone who's been into gardening knows that if you dig up in the soil, there are plenty of little critters down there that many birds love to feed on. However, because they're buried, they do pose the difficulty of how do you find them? Now, various mammals, such as these little moles, have evolved specialized limbs or other digging apparatus to get to these tasty morsels hidden beneath the ground. If we have a look at most birds, they don't really have the equipment for digging in soil. So how do they get to them? Well, various different birds have evolved this specialized foraging mechanism known as probe foraging, whereby they probe their beak into the soil or water or mud and pull out these buried prey items. There are various different birds from all over the bird family tree that make use of probe foraging today. And here are just some examples. We have a hoopoe, an oyster catcher, a curlew or a snipe, I'm not 100% sure what that one is, and an ibis. And basically what we can see is despite the fact that these birds are not closely related, they all share a similar characteristic in that their beak is long and pointed and very often curved downwards. So that's generally what the beak shape looks like of a probe foraging bird. As I said, probe foraging is a specialized method used by birds to acquire buried prey. But how do they actually find this prey? How do they know where it is? Well, there are two methods. First method is what we would call direct touch. This is used by birds such as oyster catchers, and in fact was the method believed to be used, well, we thought that all birds that were probe foraging used for a very long time. What this means is that although these birds can use some sensory cues, like maybe sound of burrowing creatures or visual cues, like little tracks on the surface of the soil that show where their prey has been, for the most part, they only know exactly where their buried prey is when it comes into direct contact with the beak. So it's a somewhat random way of finding food. It is suitable and it does work because there are many birds that only use direct touch. But today I'm going to be talking to you about a very specialized ability that some birds have known as remote touch. Aside from my fossil work, I work mainly on ibises, and they are one of the families of birds that can use remote touch. And what remote touch refers to is the bird's ability to detect prey, the exact location of their prey, before it comes into contact with the beak, even in the absence of all other sensory information. I'll explain how that works now. There are two main ways that the birds use this. So firstly, when they're hunting moving prey, so something like an earthworm that's wriggling around in the soil. So as the earthworm moves around in the substrate, this creates vibrations, mechanical vibrations in the substrate. Then we have our probe foraging bird that goes along probing its beak into the substrate. Then if the little worm moves around while this is happening, this creates vibrations, and these come into contact with the bird's beaks. And these birds have a very specialized organ at the tip of their beak, which has been very creatively named the built-up organ by ornithologists. And what this lets them do is detect those vibrations and tells them, hey, there's something down there that I can eat and where it is in relation to their beak. So this is all very well and good when they're hunting for things that are wriggling around in the soil. But as I'm sure many of us who have observed birds probing in the ground, they don't always hunt for things that move around. For example, many sandpipers and shorebirds feed on mussels or mollusks, which for the most part do not move around, so would not be producing these vibrations for the birds to detect. So how do they find these prey items? Though it is a similar mechanism, it is slightly different. So what these birds do is they actually probe very rapidly in a drilling motion, and this creates its own set of vibrations, which are then reflected off the hard shells of their stationary prey items. And it is those reflected vibrations or actually changes in pressure gradients that these birds are detecting and using to find their prey items. Well, that's very well and good for me to explain how do these things work, but how do we as scientists know of their function? So essentially what we're looking at here is a sense that we as humans do not present. So how do we know that it exists? Well, when we test senses in humans, this is quite easy. We can use various different stimuli and ask the participant what they're experiencing. This is quite different for animals. For a start, cognition is an issue. And second of all, even if we could get them to understand the test we are running, we probably would have some difficulty communicating with each other. So how is it that we do this? And I'm just going to explain to you an example from my work on hadeda ibises. They're a South African species of birds, but this is the work that was done to test remote, or very similarly work is done to test remote touch in various other species of birds. Firstly, I worked with some birds that are held captive at a sanctuary here in Cape Town. These birds, for the most part, were wild birds that were injured and then brought into the sanctuary by members of the public and were unfortunately, although the sanctuary managed to nurse them back to health so that they didn't die, they're not well enough to be re-released into the wild. So they live out the rest of their lives here in these nice big open aviaries with other birds of their species. How does our experiments work? Just to show you a basic setup, there's me with one of my study participants. So what you can see here behind me, I have a kind of plastic fence set up that's basically just like a paddock, which keeps out the ground-dwelling birds like peacocks, which also live in the aviary because they like to come and interfere with our experiments. As you can see, there is an opening there. All our birds walk in voluntarily, where they are not forced to take part in this, and I'll explain why exactly they do take part. Have our camera set up so we can film their behavior. And inside the paddock, we have these plastic trays filled with soil that the birds probe in. As I said, we train these birds how to come and forage in these soil trays. And like I said, we don't force them to do it. It's all done with positive encouragement. But unlike this photo might suggest, that's not done through compliments and thumbs up, but rather by providing them with food rewards. So buried in the soil, we have big, fat, juicy worms, which the birds absolutely love to eat. And they usually follow me around when I go in the aviaries, waiting for me to let them go in the paddock so that they can get their morning snack. So here is, again, as I said, these are the trays of the birds foraging filled with soil, and we put the worms in there that the birds have to probe out. So how do we then test their ability to use remote touch, or more specifically, test their ability to detect the vibrations that the worms are giving off? Well, firstly, we have to make sure they aren't using any other sensors to locate the worms. So we have to cut those off. How do we do that? Well, we cut off sight very simply. The worms are buried underneath the ground, and we make sure the surface of the soil is completely smooth. So there's no visual cues for them to track where the worms are. To make sure they can't hear the worms, we have a little Bluetooth speaker that plays quite annoying white noise, sort of the noise that a TV makes when it's been untuned. At the level of the speaker. This doesn't deter the birds from coming in. They don't mind the speakers at all, but it does stop them from being able to hear any sound that the worms might be making when they wriggle around. And finally, to make sure they aren't smelling the worms, we have crushed up dead worms mixed in with the soil, which means that the soil smells exactly the same as the worms, so they wouldn't be able to differentiate it based on that. Then in order to check that they are able to locate the worms just using vibrations, we test the difference between looking for dead worms and looking for live worms. And now, because when the worms are dead, they aren't giving off any vibrations because they aren't moving, and they're soft-bodied, so they can't be reflecting the vibrations back the same way that mollusks did when I showed you that example earlier. So basically, when they're dead, there's no vibrations for the birds to sense, whereas when they're alive, there are vibrations because the worms are wriggling around. And what we find is that in the birds that can use remote touch, they are much more successful at faster at finding the worms when they're able to sense the vibrations than when the worms are dead, because there's nothing for them to find them dead. And basically what I do is watch videos, something like this, of the birds poking around in these soil trays and pulling out the tasty worms. So that's one way that we test it. And probably the most simplistic way that we look at this. There are various other methods that we have used to confirm these findings, but I thought I would just explain this because it's the most sort of practical and real world understanding of it. So which living birds do we know that use remote touch? Well, there are three groups. This picture here is showing the family tree of all modern birds. And really the most important thing to note here is, as I said, there are three groups that use remote touch. It's not all probe foraging birds, as I said earlier. The three that are able to are the kiwi, ibises, and the shorebirds. And just to give you some better pictorial examples of those. So we have the kiwi, which are ground-dwelling birds, nocturnal, that live exclusively in New Zealand, really fascinating guys, and really unusual birds that are able to use remote touch. Then we have the family of birds, which is the ibises and spoonbills. These birds are found, various different species all over the world. And finally, the most diverse group that makes use of remote touch would be the scolopassant shorebirds, so things like sandpipers, godwits, snipes, curlews, woodcocks, and even the incredibly unique, beautiful, adorable, and unfortunately critically endangered spoon-billed sandpipers. So basically, there are three groups of birds that are able to use this, and within these groups, there are diverse species which make use of this really unique sensory modality to locate their prey. So how does it work in these birds? We interpret visual signals using our eyes. We interpret auditory signals using our ears. We interpret scent or olfactory signals using our noses. What do the birds use to interpret these mechanical signals or vibrational signals? As I said earlier, these birds have a specialized organ known as a bill-tip organ. And this is made up, I will now explain the various components that this is made up of. The actual receptors, little units that sense the vibrations, which we call mechanosensory receptors because they sense mechanical vibrations. In birds, they're called Herbst corpuscles. Here is just a drawing of what that looks like. It's a little fluid-filled sac with a nerve running down the center. And here I have some cross sections that have been magnified to a very high amount. These little corpuscles are about less than 1/10 of a millimeter in diameter. So they're really, really small. So it's a very high magnification of a cross section. And the bright colors are just stains that we use to visualize them. And all of the sections and bones you'll see in the slide are from hadeda ibis, which are South African species of ibis. So these mechanoreceptors, Herbst corpuscles, are embedded in high numbers in pits in the bone of the beak. So this is again cross-section of a single pit in the beak bone. You can see multiple little Herbst corpuscles within it. If we zoom out, this is a cross-section of the entire tip of the lower beak of hadeda ibis. The colors used to stain the tissues here are, again, as I say, just to help us visualize them. So bright pink. The outside is the keratin beak skin or rhamphtheca, which is the part of the bird's beak that we can see with the naked eye when you look at a bird. Then underneath that, in bright blue, we have the bone of the beak, and then the lighter blues and purples are soft tissues like nerve tissue, as well as the sensory receptors. So what this picture is showing here is you can see multiple pits at the tip of the beak with these mechanoreceptors embedded in them. And if we have a look at the bone of the beak, so in this case, the soft tissues have decayed and been removed from the bone completely. And this is just looking with the naked eye. You can see that on these, with birds that have this remote touch built-up organ, the bones of their beaks have hundreds and thousands of little holes or pits that form this very distinct clustered honeycomb pattern on the beak bones. And what we found, in fact, is that birds with a remote touch built-up organ have the highest number of pits by far than any other type of birds. Very obvious to see when you look at it, and I'll show you now. And now I'm going to show you what it looks like in most other birds, in other words, birds that do not have this type of built-up organ. So here, here I'll use an example. As I say, this is the case for most birds. Example of a gull. Sorry. This is a picture of the bone of the beak of the gull. We can see that there are a few little pits on the beak bones. However, they're not nearly as numerous as they are in the ibis. And if we have a look, sorry, voice is getting all husky. If we have a look at the soft tissues of the beak, again, the stains, the red and yellow are the keratin layer surrounding the beak. Dark blue is the bone of the beak, and then surrounding that is the soft tissue. If we zoom in on a portion of the soft tissue, we can see that there are some herpes corpuscles in the soft tissue. However, these herpes corpuscles are constrained to the soft tissue and keratin of the beak and not embedded in pits in the bone of the beak. So these pits that you see don't have any herpes corpuscles in them, whereas in the remote touches, that's where we find the herpes corpuscles. And in fact, in some birds, they don't have any herpes corpuscles in their beaks at all. The only exception to this is found in the ducks and geese. If we look at their beak bones, again, we can see there are some pits on their beak bones, and it looks like there are a few more than there are in the normal or no built organ birds. Then if we look at the soft tissues, we can see that they do actually appear to be clusters of herpes corpuscles that are joined together to form these weird little finger-like projections at the tip of the beak. And what's happening in the ducks is different to the remote touch probers, because these herpes corpuscles are actually arranged with various other sensory receptors into specialized units, which we call touch papillae, which looks something like this, sort of a little pointy finger-like shape that protrudes from the bone through the soft tissue, through the beak skin, and actually onto the outside of the surface of the beak. If you have a look at the inside surfaces of a duck's beak, even with the naked eye, you can actually see these little nodules sticking out, which represent the external parts of the touch papillae. So this is a different type of bony built-up organ. It is a bony built-up organ. And of course, anyone who's seen a duck knows that they don't probe forage, so they are not using this for remote touch. In fact, they are using it when they dabble or sort of sift through the water or very waterlogged mud to pull out small particulate matter. Returning to the bird family tree, I now want to move on to show you guys some of what our research is on our fossil birds. But first, I just need to get a little bit of technical information out of the way. As I said, this is the family tree of all modern birds. All modern birds are split into two large groups, the neognates and the paleognates. Now, the neognates make up the vast majority of birds we see today, ranging from things like ducks and chickens, all the way through to the passerines, which are the perching birds, as well as parrots and various other birds. The second group, the paleognates, which is what I'm going to be talking to you about from now on, are a much less diverse group of birds and a much more unusual group of birds. They're also sometimes known as the Raptites. So this is a breakdown of the family tree of the paleognates. And this contains birds such as the kiwi, ostrich, and rhea. And I'll go through it now just to give you all the examples. The sort of main four that most people know of, or that's most characteristic of this group, would be the ostrich from Africa, rhea from South America, and the emu and cassowary from Australia and the Pacific Islands, north of Australia. So all these four birds are massive birds that we have around us. Well, some of us have around us today. They're all ground living or ground dwelling and completely flightless, and feed mainly on vegetation and seeds, but very often just basically anything that they can pick up off the ground or off the bush. They're not particularly fussy about what they'll eat. The second group I want to talk to you about is the kiwi elephant group clade. So, as I said earlier, kiwis are one of the three groups of modern birds that are able to use remote touch probe foraging. So these little nocturnal birds from New Zealand probe in the ground to pull out invertebrates. And in fact, along with remote touch, they have a keenly developed sense of smell to also help them in locating their prey. And they're the only birds, in fact, that have their nostrils located right at the tip of their beak, instead of at the base of their beak, in other words, where the beak joins the skull, which is how all other birds are. Like I said, these birds are nocturnal, don't have very good sense of sight, and that's why they have this highly developed sense of smell as well as remote touch to locate their prey. These birds are also ground dwelling and flightless, like the other paleognates I've shown you. However, they are much, much smaller, none of them really getting any larger than knee height of an average human being. What's really curious is that genetically speaking, their closest known relatives in the paleognates are actually the elephant birds. These are an extinct group of birds that once roamed the island of Madagascar. They're the largest known bird to ever have lived. Here in this picture, you can see an example of a chicken egg next to the egg of an elephant bird. And there is an egg of an elephant bird next to the skeleton of an elephant bird itself. These birds were absolutely enormous. Again, here you can see some with hippos. They were huge. We believe, or we know that they fed in a similar way to modern day ostriches and the other Paleognaths, although perhaps being even more herbivorous than the other ones. It's just crazy to think that the closest living relative of the little kiwi in New Zealand are the extinct Madagascar elephant birds. And this just shows you just how big they were compared to an average sized human being. They were very, very big. And finally, the last group I want to talk to you about is the Tinamu and moa. Tinamu are a group of living birds. They live in South America. They're the smallest members of the Paleognaths, roughly about the size of maybe a francolin. Some of them do get a little bit larger, but again, very small for this particular group. They too are ground-dwelling birds, just like the rest of the Paleognaths, and they feed on seeds, mostly seeds, and occasionally insects that they will peck off the surface of the ground. However, where they are unique in the Paleognaths in that they are able to fly still, even though they spend the most of their time on the ground, they can fly, and many of them spend their evenings roosting in trees. Another interesting little fact about them is that here you can see various eggs of different Tinamu species, as they have these beautiful, shiny, and often multicolored eggs. Again, as with the kiwi, they have a really interesting genetic closest relative, and that is the moa. And these are the giant extinct birds from New Zealand. The moa, like the elephant bird, went extinct in the last few hundred years thanks to human activity. And although the moa are nowhere as near as large as the elephant birds, they are still much, much larger than their closest living relatives, the Tinamu. Now that I've introduced you to all the members of the modern Paleognaths, and point out, I have shown some of them, but just show you exactly how weird evolution has been in this group. So here's just a schematic of the size differences. We have the two extinct members here on the left, and then the other living groups here compared to human being for size. But this chart shows just how diverse they are in terms of their geography, being found all over the world. However, it is important to note that they are only found in the southern hemisphere, these modern Paleognaths. If we look at their family tree and how closely related, we can see that this smaller size has evolved various times in the Tinamu and Kiwi. So it's not found in all of the Paleognaths, and it's not found in these two species, despite being both similar in size, are not closely related compared to the other Paleognaths. We also have this weird lack of flight in all of the Paleognaths, except for the Tinamu, which seem to have reacquired the ability to fly. And finally, as I said earlier, the Kiwi are the only species that use probe foraging to look for food. And it is this particular trend that we will be talking about further on. So now I'm going to move on to the curious case of the non-probing Paleognaths beaks, because as the talk suggested, I am very interested in beaks and their ability to sense vibrations. So the Kiwi are the only members that probe forage. Here's a nice picture of a Kiwi probing for things on a beach. And if we have a look, and so they are the only members of this group that would have a need for using remote touch. Here again is a Kiwi skull. You can see lots of little pits on the beak bone tip. And again, if we look at the soft tissue, they have those receptors inside the pits of their bones. So we wouldn't expect to find this anywhere else in the Paleognaths because they don't have a need for it. But let's take a look. And I'm going to show you an example from the Tinamu. They're the other little small Paleognaths. So if we have a look at their skulls and their beak bones, we can see that they seem to have quite a high number of pits on the tip of their beak bones, which is weird. Why would they have this? Although it's not quite to the same extent that the remote touch probing birds do, like this ibis, it is still really high and much higher than that example I showed you earlier of the gull. So what's going on? We then have a look at the soft tissues inside these pits of the Tinamu. Here we can see again, there's lots of those little pits. And if we zoom in, we can see that, hey, they actually have Herb's corpuscles in these pits, which is the same as the tissue arrangement in the remote touch probing birds, indicating that the Tinamu have a built-up organ. And this is very curious because, as I said earlier, these birds do not probe forage for food and have no need for a remote touch filter organ. So why am I showing you this? Are the Tinamu just a weird aberration in the Paleognates? Here are just some pictures of all the modern Paleognate skulls. And if we have a look at their beak bones, no, the Tinamu are not unusual in the Paleognates. In fact, all of the Paleognates have this high number of pitting in their beak bones, despite, obviously, aside from the kiwi, none of them using or needing remote touch. Even the moa and elephant bird, which are extinct, show this high number of pitting on their beak bones. And obviously, we can't look at the soft tissues for the moa and elephant bird because we don't have any of them preserved in the fossil record. But in all the living or extant Paleognates, we have found the soft tissue organizations indicating that they all have some type of built-up organ. But why? Why on earth would they need a remote touch probing built-up organ? None of the other Paleognates, aside from the kiwi, make use of probe foraging, and in fact, most of them are very unpicky about what they will pick up and eat off the ground. Another study once suggested that potentially they might be using this built-up organ for another purpose. This is something that can happen. Things can be used for different functions in various organisms. However, if we have a look at the receptors in the built-up organ, there are no other types of receptors. I think one possible suggestion was that maybe they were using their built-up organ to judge the temperature of their eggs in the nest, which could be useful because you want to keep your eggs at the right temperature. But there are no thermoreceptors or chemoreceptors or any other type of receptors in the built-up organ. All it can sense is mechanical vibrations. And these birds don't need that. Furthermore, these birds have actually lost the specialized areas of their brains, which allow them to interpret this tactile information. So they're not able to interpret the information that they would get from this built-up organ. They don't need it. They can't use it. So why do they have it? Well, our idea is the fact that it's present in all of the Paleognates could indicate that it could be a vestigial organ passed down from a common ancestor who used to be able to use remote touch probe foraging. And we wanted to investigate that. In order to do this, we needed a candidate or group of candidates of very ancient Paleognates to try and see if we could find if they were able to use remote touch probe foraging. The group we decided to look at as a possibility are the lithornithids. Lithornithid means stone bird. And this is simply just as a just refers to their presence in the fossil stone record. These birds are known as basal Paleognates, and here on the left we can see an artist's representation of what they might have looked like, and here is just a fossil of a lithornithid preserved in stone. As you can see from the picture, they were fairly long-legged, robust little birds, roughly the size of a large chicken, maybe a little bit bigger, a little bit smaller, depending on the species, fairly long necks and longish beaks. However, like the Tinamu, we know that they were, based on their wing structure and various other parts of their morphology, we know that they were able to fly. And actually, this has been a well-established fact, not just from the lithornithids, from various Paleognate fossils and genetic information, that all modern Paleognates evolved from a previous ancestor that could fly. Their position in the family tree makes them particularly useful. So this is the main family tree I showed you earlier with the modern Paleognates, and the lithornithids fall as a sister group to these modern Paleognates. So they're not the ancestor of all Paleognates, but they would have been around either just before or at a similar time to that ancestor, and are equally related to all modern Paleognates. This group evolved in the Cretaceous period, so between 166 million years ago. And that was at a time, one of the most famous times, I think, in prehistoric history, when some of the biggest dinosaurs walked the earth, as well as pterosaurs and various other strange creatures which we no longer have around with us today. However, as many of us know, the Cretaceous was not to last, and due to catastrophic environmental events and changes in climate and geography, all of the large non-avian dinosaurs went extinct. And I say non-avian dinosaurs because, as we know, birds are the only living type of dinosaurs that remains behind today, having evolved from dinosaurs millions and millions of years ago. And if you want to know more about that, you should check in for Anusha's talk next Monday, because she will be explaining that. It's a fascinating but very broad topic, which I don't have time to cover today. Another thing we know, however, is that some dinosaurs did survive this catastrophe, and that is birds. Namely, we know that mostly ground-dwelling birds survived best, and the lithornithids were among these birds that managed to survive and continue and diversify. However, they did go extinct around 40 million years ago, and here's just a really beautifully preserved fossil of a lithornithid. Again, you can see the long legs of it preserved there. Various different studies indicate, based on their morphology, that they were probably semi-aquatic waders living close to water bodies and dwelling on the ground and perching in trees at night. So all of this, and especially their basal position in the Paleogenes family tree, makes them an excellent example for us to use. However, before we can even think of using them, we have to study their beaks, which means we need really well-preserved specimens of their beaks, because I showed you earlier, the little pits on the bone are tiny and really delicate. And we knew the odds of being able to find specimens like that from the fossil record were almost zero, because fossil record is extremely destructive and you can't decide that you want to study something. You kind of just have to make do with what there is, because fossils are relatively so rare. However, in this case, we were extremely, extremely lucky. So here I have some pictures of various specimens from two species of lithornithids, both from North America, and both over 45 million years old. And as you can see, their beak bones have been exquisitely preserved. If you have a look at the tips of the beaks, we can see those foramina, which are present on all birds' beaks. But we also can see that it seems like they do have a higher number of foramina, much like the rest of the Paleogenes, and potentially even as many as the remote touch probe-foraging birds. So what we need to figure out is, based on their beak bones, did they have a bony built-up organ, and were they able to use remote touch? I'm not going to go too into details on the methods or the very specific results. I'll just show you a couple of results and give you a brief overview of what we did. So firstly, to justify using the little pits on the beak bones as a proxy or a kind of a measure of the built-up organ, we had to make sure that they were representative of the soft tissue structures underlying them. In other words, that we could link the two, and we were able to, basically showing that the very high numbers of bony pits on the beaks indicated that there would be herpes corpuscles and soft tissue arrangements of the built-up organ in the beak. And from this, we had 52 species from various different families. A skeletal sample of living birds, we had over 250 families from a huge coverage for a sample like this, and all 41 orders or groups of modern birds, living birds. We also had the fossil Paleogenes. We had 4 lithornithid species that we looked at, as well as 1 elephant bird and 1 moa. And the general type of measures that we took were, we looked at beak length, the number of bony pits on the tip region of the beak. Also looked at the average nearest neighbor distance, which just refers to how close the pits are together. And finally, we looked at a ratio of beak to skull length, which just refers to long and pointy or short and stubby the beak is with regard to the skull. So just to show you a couple of graphs. So don't worry too much here about the values on the X and Y axes. Those are PC values, which just refer to a type of modeling that we use to display this type of data. What's really important are the actual measures that we're looking at on this graph. The two measures are the number of pits on the beak bones, as well as the average nearest neighbor distance between the pits. In other words, how closely together the pits are. So if I add the data, so each point on the graph represents a species of birds, and the triangles represent fossil specimens, fossil species. Firstly, I just want to highlight the 2 shaded areas. We have the gray shaded area and the green shaded area. So the green shaded area is birds with some type of bony built-up organ. So the ducks, the remote touches, and the Paleogenes. Then in gray, we have all the other living birds, which we know don't have any type of built-up organ. And basically what our data here is showing is that we can differentiate just looking at the bone structures between birds with a bony built-up organ and birds without. This region is just showing a zoomed in part of this graph, just so you can see more detail. And the colors of the dots here are showing different categories. So blue would be the ducks and geese, purple would be the remote touch foragers, green would be the other paleognates, so the non-kiwi, non-probing paleognates. And then the red triangles are the lithornithid specimens. So looking at the extant birds, just based on this data, we cannot separate these groups out in terms of the function. We can separate that they have a bony built-up organ versus not a bony built-up organ. But we can't say exactly the function of it just yet. However, what we can see from this graph is that the lithornithids are falling definitely as separate from the other birds without a bony built-up organ, and falling well within the group of birds that have some type of bony built-up organ. Don't worry, this is the last graph I'm showing tonight, but it is probably the most important one. So all you need to know, on the x-axis here, we have kind of a measure of how long and pointy the beak is. So on the left-hand side would be sort of very short, stubby beaks, and on the right would be long, thin beaks. And then on the y-axis just refers to the number of pits on the beak bones. Looking at the data, again, each point is a different species of birds. This purple shaded region is the remote touch probing birds, which have long, thin beaks and lots of pits on their beaks. And then in the green region, we have the non-probing paleognates. Obviously the kiwi, which are paleognates, are part of the remote touch probing birds. But the non-probing paleognates all have shorter, stubbier beaks than the remote touches, and generally less or not quite as extremely high numbers of pits on their beak bones. Really, what the most important thing is with this graph is that these two groups are separate. There's absolutely no overlap in terms of their beak morphology, length, and number of pitting between the purple and the green group. However, then when we added the data from the lithornithids, these four triangles are for the lithornithid specimens, which we could count the number of pits on. And then the two vertical dotted lines refer to two other lithornithid specimens, which unfortunately their beaks were too badly eroded for us to count the pits. However, were able to measure their beak shape. So we've plotted that here on the graph. And we know based from the other specimens that we did count the pits, the kind of range that they would fit in. And this allowed us to shade a possible area of what the lithornithid group would have taken up relative to these other two groups. And what this data shows is that from the ones that were measured, that we were actually able to measure, the species either fall in the group of remote touch probing birds or as intermediate between the remote touchers and the other paleognates. And what is also important to note is that while for the living birds, we were able to select absolutely exquisitely, perfect specimens without any damage. Unfortunately, as I said, because of the fossil record, there is some damage to the beaks, even for the beautifully preserved ones. And so we only counted pits where we were 100% sure that there was a pit there. So actually the pit counts for all of these will be higher, which pushes them even further away, which would push them even further away from the non-probing paleognates. But this is just a very conservative measure. They would actually be higher up. So what can we say from this about remote touch in the lithornithids? Well, clearly they had some type of bony built-up organ, as would be expected, based on their grouping with the paleognates, and just looking at the beak bones, it's very clear to see that. The beak morphology shows that their built-up organs were distinct from that of the ducks and geese. I didn't show that data here, but various other morphologies of their beaks, as well as just their general morphology, we know they didn't have the scabbling type of built-up organ. And I've also shown how we have shown they were distinct from the non-probing paleognates, so the other paleognates. So depending on the species, their beaks morphology was either more like that of the remote touch probe foraging birds or an intermediate of the remote touch probers and the other paleognates. And so what we've concluded from this is that some, if not all, species of lithornithids were able to use remote touch probe foraging. And based on what we know from previous research on the evolution of the lithornithids, in that they first evolved during the late Cretaceous period, indicates that remote touch had evolved as long ago as then, potentially even earlier, which I'll get to just now. How does this impact our hypothesis on the vestigial built-up organ in paleognate birds? Well, as I say, the lithornithids having this functional remote touch built-up organ explains the case in all the other non-probing paleognates as a vestigial organ that was inherited from a previous common ancestor. There are many examples of this type of retention of a useless organ or body part in animal record. I'll just give one other well-studied and well-known example. So modern-day whales, as we know, if you have a look at them, they just have their front limbs evolved into flippers and they have no visible hind limbs and really no need for them or for the pelvis which attaches to them. However, many of them still have this vestigial pelvis structure in their skeleton, even though it's completely useless and isn't attached to their vertebrae anymore even. And we can see how this has changed in the fossil record when their old ancestors who used to live on land, who had normal pelvises that were used for walking and have slowly been lost, but still retained, well, not lost, have slowly changed, but have still been retained in modern day whales. And this would be the same in the paleognates. So they still have this built-up organ, but it's not quite as extensive on the beak, and they've lost the corresponding brain regions required to interpret information from it. So it's kind of just there. And this happens very often in nature when this type of structure on the body doesn't pose any kind of cost to the birds. It doesn't cost them anything to have this built-up organ. So there's no drive for them to actually lose it. Looking at the evolution within the paleognates, so how did it happen that in the modern birds, despite the fact that their previous common ancestor was a remote touch prober, how come only the kiwis have that now today? So in answering evolutionary questions like this, we try and look for what we call the most parsimonious pathway, which is really just the most simple, or the evolutionary pathway that would take the least number of steps. In this case, that would mean a loss in remote touch between the lithornithids and all the modern day paleognates, and then a regaining of it in the kiwi after they split from their common ancestor, which they shared with the elephant bird. However, as I showed you earlier, evolution in the paleognates has taken some very weird pathways regarding their loss and regaining of flight, size differences, as well as foraging niches and other things. So it may very well be possible that there have been multiple losses and gains in the ability to use remote touch throughout the paleognate lineage. And while our study hasn't been able to show that, in order to do this, we would need to look at fossil samples of, unfortunately, as of yet undiscovered paleognates, or looking at the genetic pathways that underpin the development of the built-up organ or the associated brain regions required for using remote touch. So that's work that still needs to be done, but just to show you kind of, there are various possibilities of how this could have happened in the kiwi. As I said, I've just presented you with a potential question. Well, research, being as it is, generally tends to lead to more questions than it does answers. So here I'm just going to show you some of our conclusions and questions that have led from our research regarding the evolution of remote touch in birds. One possibility may be that it evolved for the first time in paleognates after they split from the other group, the neognates. And it could even be possible that the lithornithids were the earliest possible example of remote touch in all birds. However, something interesting to note is that there are various similarities between the duck type and the paleognate type built-up organs. So here are just some pictures. Paleognates, the rhea's beak bones, and then a duck's beak bones. And as you can see, sort of general layout of the pitting on the beak bones, as well as the divisions of the different parts of the bones, are quite similar between these two groups. Why is this interesting? Well, if we have a look at where the ducks sit in the neognates, so we have a big group of paleognates, sister to the neognates, and the ducks sit with the group of birds, the galliforms, which are things like pheasants and chickens, right at the base of the neognates. So very, very close to, to the base of the neognate clade. And based on this basal position and the sister group of Paleognates, this could indicate, and this very similar built-up organs between the two groups, this could actually indicate that these two types of built-up organs evolved from another type of built-up organ, whether it was used for dabbling or remote touch or something completely different, we don't know yet, or an even earlier ancestor of all modern birds that we know today, which would be really, really cool. And although no conclusions have come yet, there is quite a lot of work ongoing, both by us and other groups looking into this. And we are very excited to hopefully soon be able to read and talk about that. But we believe that this is probably the more likely case that happened than that it evolved for the first time in Paleognates. But for now, that is just conjecture, but it's nice just to be able to share our theories. Something else interesting is that similar structures may have evolved separately in crocodiles. So crocodiles evolved separately from birds and dinosaurs, also a very, very ancient group of animals. Here I have a picture of a skull of a crocodile. And you can see along the jaw bones, they have lots of these little pits, similar to the way that the birds have lots of pits in their beak bones, and birds' beak bones are just a specialized type of jawbone without teeth, obviously. We don't yet know, it hasn't yet been published, although we think it is being worked on by another group, as to whether there are mechanoreceptors embedded in those pits, but hopefully we'll find out soon. What we do know is that crocodiles possess a very similar sense to remote touch, in that they are able to detect mechanical vibrations in the water that they swim in, and they use this, to hunt for fish and also potentially for communication between them. And as I say, this isn't exactly like remote touch, but it is a sensory system that uses a lot of the same principles, which is really interesting, as it shows it's kind of an analog to remote touch in a completely separate group of animals. However, what we find really exciting is that there is increasing evidence that some of the non-avian theropods, and the theropods are this group of quite well-known dinosaurs, including things like T-Rex and raptors from Jurassic Park fame. And this is actually the group that gave rise to modern birds. And there is more and more evidence being published from other researchers showing that some of them seem to have specialized sensory structures on the distal, or kind of the front portions of their snouts. One example that was published recently was looking at the snouts of a type of dinosaur called Neovenatus celeri. This is a theropod from what is now Britain. And as you can see, it's quite a large theropod. And so they found what looks like there might be some tactile or sensory receptors on their snouts. Obviously, no one is suggesting that this animal was probing in the ground like a modern day ibis. And we're not 100% sure yet with this particular group what they were using this for. However, more sort of well-studied or better hypothesized studies were looking at a group called the Spinosaurs. And this is particularly looking at one species from Egypt. These are massive dinosaurs, as you can see, also from the Cretaceous period. What this study did is having a look at their snouts. You can see again, a high number of foramina or pits on their jaw bones, potentially indicating a sensory specialization. So these very large carnivorous dinosaurs also gained some fame in the Jurassic Park movies. I think in the second movie, they were the big bad in there. These large dinosaurs that had these like sail-like structures on their backs with long pointed snouts. For a long time, it has been accepted that they lived near or in water and hunted on fish and other large swimming animals, potentially even crocodiles. And many people for a long time believed that they would stand in the shallows and hunt visually, looking down and catching prey as it swam underneath them. However, more and more studies of their general morphology have indicated that they may well have had a much more aquatic lifestyle and had swum around more like a crocodile than just standing in the shallows and hunting. And a very recent paper that came out a couple of months ago, also looking at their overall morphology, has suggested that in fact, no, they weren't swimming, and they might have been wading, as was said before, but instead of standing above the water and looking down at their prey, they had submerged their snouts in the water to hunt for fish in that way. And while this study compared it to something like herons and storks do, which do not have the ability to detect vibrations, they just hold their beak open until something actually touches the beak. A fish swims through and touches the beak, and then they snap it closed. But it could also potentially be in a similar fashion to things like ibises and spoonbills, which do this and are able to use remote touch. However, the authors of this paper did not discuss their results in respect to this, or in respect to the fact that very interesting sensory structures have been described by other authors in this family of dinosaurs. But we do believe that this is work that is ongoing, and we're really excited to hopefully see where it goes further. There is still a lot of conjecture and a lot of contention within the paleontological community as to what's going on with spinosaurs, but this is definitely an area that we're very excited to watch and see what comes out of it. As I said, there are numerous different examples of these theropods maybe having some kind of sensory specialization on their snouts. Again, I want to stress, no one is suggesting that they were probing in the ground like birds. We're probably using it more similarly to the way crocodiles use their sensory structure. However, why this is exciting is, even though it is different to remote touch in birds, because we know birds evolved from various theropod dinosaurs, it could very well maybe that these specialized sensory structures in the theropods may have evolved into the highly specialized remote touch built-up organs in birds, and provided kind of the morphological backbone and genetic framework for this to develop in birds, which is really exciting because it would mean one of the most specialized feeding mechanisms that we see in modern birds evolved from the dinosaurs. But as I say, this is all still conjecture, and hopefully in the next few years or decade, we will have more answers. As I'm sure you've realized in these past couple of slides, I've been presenting many questions and not many answers to you. And that is just one of the beautiful things about science. As I said earlier, answers to questions just provide more questions. And that's what keeps driving things forward and keeps scientists like us both employed and intrigued and wanting to carry on. And I always like to present these questions, even at talks like this, just to kind of give people the idea that science is never a fixed or a complete topic. We're always finding new ideas and new reasons, and any scientist who tells you that they have a theory that covers everything or the answers to everything, whether it's a bird, whether it's an ornithologist or an astronomist, big question marks. But yeah, it's always good to have facts and strong theories, but yeah. So what are some of the takeaway messages, I guess, really from our research? Well, we're pretty confident that that our data show the lithornithids were able to use remote touch, and that this explains the vestigial state in all of the modern paleognates. And even then, as I said earlier, it might be that the lithornithids are the earliest example of remote touch in birds. So even if it didn't evolve before then, or even in the very earliest birds, it still means that it is one of the oldest sensory specializations for foraging in birds that we know of today. And we really liked, so the New York Times wrote about our paper, and they called this an unusual bird superpower, which we like because obviously it's not a superpower, it did evolve naturally, but it's a sensory mobility that we as humans don't have, which I guess is what makes it super. And it's just so old, and it's crazy to think that something so sophisticated and unique to birds existed as long a time ago as when the gigantic dinosaurs ruled the planet. Finally, I'd just like to thank the following people and organizations for hosting me and assisting me in this research. Huge thanks to our funders, the Center of Excellence in Paleosciences, the Fitz Institute, and the National Research Foundation. Massive thanks to my two supervisors and co-authors, Anusha and Susie, for just amazing mentors, and just so grateful to them for all of the support and guidance they've given me, and for allowing me to do the crazy things we proposed over the last few years. Finally, again, huge thanks to Derek for inviting me to come and talk at Learn the Birds. I think it's such an incredible initiative, and I'm so happy to be a part of it. And thanks to all of you that came along today. It was really great to have you here. And yeah, if you want to ask any questions that I can't answer today, please feel free to send me an email. I'm also on Twitter. My username is Carla du Toit. Not super active on Twitter, but I will answer anything anyone sends me there, and do post any of our new research up there. Thank you.
Thank you so much, Carla. This was an amazing presentation. I'm just mind-boggled, and you know, I'm a biologist by trade, and you know, learning about this is always something that just fascinates me, and this is really great. So I think we are 29 people in the room. I'm going to just ask if people have some questions, if they would just type them into the chat. I'm just looking up through the chat now to see if there are any questions. Mostly it's just people commenting.
Oh, I see. Yeah.
Carry on.
No, I saw one person ask why the elephant bird and moa became extinct. So yeah, they similar fate to the dodo. They went extinct due to human culling and activity, and introduction of livestock and other predators, unfortunately. We know that the moa went extinct in the 17th century, late 17th century. The elephant bird is less clear exactly when, but it may have been as early as 1,000 years ago to about 1500. So somewhere over 500 years ago, they went extinct.
So I'm allowing people to unmute themselves because a lot of people have left the room, and I think it will be manageable. So if you have another question for Carla, just unmute yourself and ask it.
Oh, I've seen one quickly. There's how do penguins fit into this? That's a really interesting question. I'm busy finishing my PhD in the next couple of months, so things are quite hectic here, but I'm going to be moving on to working on seabirds. And it seems like there's some interesting developments in their beak bones, but we haven't dug into it deeply enough yet. They are sort of closely related to the ibises, not really, but kind of much closer than any of the other groups that I spoke of. But yeah, so unfortunately I can't say more about that now, but we will hopefully be publishing on that soon. Sorry.
Anything else, folks? Hillary, I see you have unmuted. You have a question.
No, I was just engaging because I asked about penguins.
Cool.
Anybody else? Someone's asking about a recording. I won't put you on the spot. I've sent you an email about a possible use of a recording.
Oh, you're welcome to distribute. As I said, this is all publicly accessible information. So yeah, sure.
Okay, in that case, we'll put you on our YouTube channel.
Cool. Oh, hello. See another histologist. I'm unusual in my lab. Everyone in my lab on campus works on bone histology, and I work on soft tissue histology and actually a lot of behavior, so alive birds, which makes me a bit weird, but great. Yeah, be interested to see histology.
If you look underneath the Kathy, if you look underneath Carla's profile on the Learn the Birds website, there's a bunch of links there that link to her websites and Twitter and those things. Okay, I think that seems to be all of the questions. I want to just thank everybody from coming. We've got 23 people left. You had 40 people here at the start, so that's good for a scientific talk. That's good. Mostly we do birding tours and things. And so I think that's a really good turnout. And thanks everybody for coming. And hopefully we'll see many of you again on Monday at 1300 South African time, and again at 1900 South African time on Monday, which is International Women's Day. So thanks again, and thanks so much, Carla.
Sure. Thanks, Derek.
Take care and have a good evening.
You too. Cheers. Bye.